It is the year 1957. A paper titled An Experimental Study of Interaction Between Genetic Drift and Natural Selection has been published by the authors Dobzhansky and Pawlowski at Columbia University. Dobzhansky, an established and well-known geneticist, has inadvertently intensified the chaos present in the genetics community by seeking experimental evidence of natural selection and genetic drift. Throughout the 20th century, there had been constant conflict between the evolutionary importance of genetic drift and natural selection. Some scientists believed that natural selection was all-important, dismissing the idea of drift. Others, such as Dobzhansky and Pawlowski, believed that genetic drift worked in conjunction with natural selection. In the manuscript, the authors identified two major opinions regarding drift. 1. As only a descriptor for differences in organisms with unexplained origins, or 2. That natural selection and drift are alternatives, either selection or drift. Dobzhansky did not regard this second definition to be logical, thus designed an experiment to test for interactions between selection and drift. Random genetic drift encompasses all of the variations in genetic frequencies which are indeterminate in direction. A preliminary experiment was set up involving Drosophila subscura. Wild-type flies with different chromosomal gene arrangements on their third chromosomes were collected. Curiously, chromosomal heterozygotes bred from flies of different geographic regions gave varied fitness and frequency results that were not uniform across replicate trials, unlike those from flies of the same locality. Race hybridization results in an explosion of genetic variability. The possible gene combinations exceeds the numbers of flies. Therefore, natural selection would promote genotypes with high adaptive values, but it was up to chance which genotypes would appear in the populations. The scientists used this knowledge to design their main experiments. The main experiment used flies with variations on their third chromosomes, collected from geographically different regions. Flies collected were either homozygous for the Pike's Peak or homozygous for the Arrowhead gene arrangements. Foundation stock was prepared by crossing the Pike's Peak and Arrowhead strains, creating F1 and F2 heterozygotes of 50-50 Pike's Peak and Arrowhead. A total of 20 populations, 10 small populations derived from founders of 20 F2 flies, and 10 large populations derived from 4,000 F2 founders, were kept in population cages of uniform condition for 19 generations. All populations were allowed to fluctuate and expand within the carrying capacity of the population cages. Periodically, chromosomes in the salivary glands, having high visibility, were taken from larvae and analyzed. By 19 generations, the frequency of Pike's Peak had decreased in both large and small founder populations, with small founder populations showing significantly greater heterogeneity. Dobzhansky and Pawlowski concluded that 1. Greater heterogeneity is due to differences in magnitude of the founder generation of populations. 2. In geographically mixed populations, the selection of the chromosomal gene arrangements is dependent on the polygenic genetic background, which is highly variable due to recombination. Here, genetic drift becomes important. 3. Hybridization led to highly variable gene pools, of which random drift determined what would be included in the gene pools of founder generations, and natural selection drove the divergent results in different populations. The smaller the population size, the more genetic drift impacted the population, resulting in greater deviations and fluctuations across generations. The results from this paper were significant for the scientific community, providing experimental evidence for several principles. Firstly, the experiment is largely believed to have verified Mayer's founder principle, as it manipulated the founder effect while controlling the variables of population size and selection pressures. However, the relevance of this research towards natural populations where founder events derived from a single locality have been questioned. Secondly, the manuscript is recognized for having shown experimentally the Fisher-Haldane-Wright theory by testing the effects of genetic drift in combination with natural selection. However, some geneticists at the time were highly skeptical about the importance of genetic drift. Ford, vocal against genetic drift, especially his understanding of Wright's views, only believed that natural selection created divergence in populations, and if drift was in play, divergence should be less stable in smaller populations than larger ones. This paper provided one of the earliest connections between experimental evolution and genetics, allowing evolution to become an experimental discipline. The experiments provided evidence for the interaction of genetic drift with natural selection, an idea that had been widely criticized and doubted, as well as providing experimental evidence for further theories.